Today we'll continue our discussion regarding the biological importance of water. Now there are four physical properties of water, namely vapor pressure, boiling point, melting point and osmotic pressure, which are collectively called the colligative properties. Because if the water contains any solute, then the solute will affect these four properties in a similar way. Okay, and this effect is dependent upon the fact that the concentration of water is lower in the solution compared to the pure water. Now this effect of solutes on the properties of water does not depend on the chemical nature of the solute. It only depends on the number of solute particles present in a given amount of water. For example, if you take a non-dissociating solute like glucose, it doesn't dissociate. On the other hand, if you compare the effect of sodium chloride, which dissociates completely to generate two ions, that is two particles, then the same amount of sodium chloride will give twice the effect of the same amount of glucose. Now, water molecule always has a tendency to move from a region of higher concentration to that of lower concentration, similar to the diffusion process. This is governed by the second law of thermodynamics, that is, any system wants to go to more randomness or disorder, that is, increase in its entropy. Now, if two aqueous solutions are separated by a semi-permeable membrane, that is a membrane that allows the passage of water molecules only or solvent molecules in general, but not the solute molecules, then water molecules will diffuse from the region of higher concentration to that of lower concentration, and in this way it would produce a force which is called osmotic pressure. Now there is a simple experimental design to determine this osmotic pressure. Like you can see in this setup, we have kept pure water in this beaker and we have dipped a tube inside. This tube is sealed in this end with a semi-permeable membrane, and we have filled this tube with a solution. Okay, and the level of the solution initially is kept at the same level with the water in the beaker. Now with time, water molecules will pass through the semi-permeable membrane and enter the tube, resulting in increase in the height of the solution along the tube. Now this will continue, there is a flow of water molecules will continue till this flow is exactly counterbalanced by the gravitational force that this column of liquid exerts on that flow of water. Then it will stop. Now if we allow a force on this water level, or I should say solution level, then at a certain force we will be able to move the water in such a way that now the level of the solution goes back to the original one. Then this amount of force is a measure of osmotic pressure, and we symbolize it as pi. The Van 't Hoff's equation describes the mathematical expression of pi equal to ICRT, where R is the universal gas constant, T is absolute temperature. The product IC is called the osmolarity of the solution, where I is the Van 't Hoff factor, which is the extent to which the solute dissociates into two or more ionic species, and C is the molar concentration of the solute. Now for strong electrolytes like sodium chloride in a dilute solution dissociates completely to two particles, that is two ions here, Na plus and Cl minus. Effectively I here will be 2. On the other hand, for a non-ionizing solute like say glucose, which will never dissociate, I will be considered always as 1. Now if the solution contains several, like n number of solutes, then the overall osmotic pressure is the sum of the osmotic pressures of all the individual solutes taken together. Like the equation will be pi equal to RT in the bracket the summation of all the osmolarities of the individual solutes.
Sir, does semi-permeable membrane exist in biological systems?
Definitely. We'll come to that. So, osmosis, that is the movement of water across a semi-permeable membrane driven by osmotic pressure. That is the difference of osmotic pressure between the two solutions across the semi-permeable membrane is one of the most important factors of the cellular properties. That means plasma membrane, there is a biological membrane. It is special in the sense that it allows the passage of water molecules much more compared to the other molecules, any other solutes, whether it's a small molecule or a bigger molecule or ions or even macromolecules. It will prefer water much more than the rest because it has some special protein channels called aquaporins through which water can pass to the either side, inside the cell or outside the cell to a large extent. So effectively we can consider plasma membrane or biological membrane as a semi-permeable membrane. Now a cell which is surrounded by a plasma membrane can have three different situations regarding the osmolarity of the surrounding medium. In the simplest case, the osmolarity of the intracellular fluid, which is equal to the extracellular fluid, in that case, since there is no difference in osmotic pressure, there will be no net movement of water across the plasma membrane. So the cell will be stable. But if the cell is put in a hypertonic solution where the osmolarity is much higher in the surrounding medium, then osmotic pressure will drive the flow of water from the cell to the extracellular material. That means the cell will shrink, which is not good for the cell. Even more disastrous effect is when a cell is in a hypotonic solution. That is, the osmolarity in the surrounding medium is much lower than that within the cell. In that case, osmotic pressure will drive flow of water within the cell from the extracellular fluid. This will result in swelling of the cell, and eventually, if this continues, will lead to cell lysis. This is called osmotic lysis. Now, normally in the natural environment, a cell encounters, in most of the cases, hypotonic solution. So there is always a tendency of the cell to undergo osmotic lysis. The cell must have mechanisms to prevent this osmotic lysis, and there are several mechanisms actually. For example, in bacteria and plants, the plasma membrane is further surrounded by a sort of non-expandable cell wall, which is sufficiently rigid to resist the osmotic pressure and thereby prevent osmotic lysis. Then again, freshwater protists, which usually live in a highly hypotonic environment, they have a special mechanism, a special organelle called contractile vacuole that always pumps water out of the cell. So a lot of water comes in the cell as well as a lot of water goes out. So effectively the osmotic lysis is avoided. In multicellular animals, they have a different strategy. They usually keep the osmolarity of blood plasma and extracellular fluid at a similar level with that of intracellular fluid. Consequently, there is a balance of osmolarity and that avoids osmotic lysis. Say in blood plasma, there is a lot of proteins, for example albumin and many other proteins, which increases the osmolarity of blood plasma to the level of the osmolarity of intracellular fluid. Now cells also pump out huge amount of sodium ions and many other ions into the extracellular fluid to lower its own osmolarity and match it with that of extracellular fluid. In this way it can balance the osmotic pressure and prevent osmotic lysis. Now let us move on to a different concept. This intracellular as well as the extracellular fluid, they need to maintain a constant pH. The reason is that you know that biological system, they depend on enzymes and each enzyme has its own pH optimum. Now, in most of the cases, this pH optimum in biological system is close to 7. Now, if there is a gross deviation from 7, then the enzymes will lose their activity and the cell will die. So, cells must have some mechanism to prevent the deviation from the regular pH. There are a lot of processes going on within and outside the cell that will shift the pH from 7, either lower it or increase it. But cells have buffer systems to prevent the change in pH. Okay. For example, the cytosol. Cytosol contains large amount of proteins. Proteins, they are composed of amino acids, and there are different types of amino acid side chain residues. Some of them are weak bases, some of them are weak acids. Hence, those side chains, they can act as buffer systems. Here, the example of histidine is shown. Histidine can exist in two ionic forms. One is a protonated form and another is a deprotonated form. So, this form is a proton donor and this is the proton acceptor. This protonated form has a pKa of 6.0. Now, the biological pH we know is close to 7 in most of the cases. So, the buffering range of histidine is pKa plus minus 1, that is 5 to 7, which is good enough to act as a buffer system for the cell. Many other small molecules, like many different metabolites such as the nucleotides, ATP and many others, they can exist in different ionizable forms, and consequently they can also act as buffers. Some special mechanisms are there for some special organelles and unique extracellular compartments. Usually they accumulate high concentrations of certain compounds that increases their buffer capacity. For example, in plant cell vacuoles, a lot of organic acids are there to act as buffer. Then in urine, it is buffered by a significant amount of ammonia present within it. There are two very important buffer systems that exist in different organisms. That is their phosphate buffer system and bicarbonate buffer system. In the phosphate buffer system, which usually acts in the cytosol as well as in the extracellular fluid, this H2PO4 minus, this species act as proton donor, and this HPO4 2 minus act as proton acceptor. The pKa of H2PO4 minus is 6.86. Consequently, it's a good buffer between the range of pH 5.9 and 7.9. Now, say in mammals, for example, the extracellular fluids and the cytosol as well as different cytosolic compartments, they have a pH within the range 6.9 to 7.4, which lies well within the buffering capacity of the phosphate buffer system. Then the bicarbonate buffer system, which usually buffers the blood plasma. This is a little bit more complicated because it has two phases. Some components, they operate in the aqueous phase, like blood in the capillaries, as well as some component they act in the gaseous phase, in the lung air space. See, here you have the bicarbonate ion, which accepts a proton to form carbonic acid. This is a reversible reaction. Now, this carbonic acid then can lose water molecule to form carbon dioxide. This carbon dioxide is a dissolved carbon dioxide. Now, this dissolved carbon dioxide again is in equilibrium with the carbon dioxide present in the gas phase, in the lung air space. All these equilibrium, they are in series and they can act as buffer system. For example, say if the blood pH drops, say during exercise when lactic acid is produced, it will increase the H plus concentration in the blood. Then H plus will combine with the bicarbonate to form carbonic acid. Carbonic acid then will lose some water to form dissolved carbon dioxide, and this excess dissolved carbon dioxide will be released in the gas phase to increase the carbon dioxide level in the gas phase, in the lung air space, and this extra gaseous carbon dioxide will be exhaled out, and thus the blood pH is regulated. On the other hand, there are some mechanisms that will increase the blood pH. That is, it will consume the protons in the blood and thereby increase the pH. For example, when proteins are catabolized, that generates ammonia, and this ammonia will consume the H plus from the blood, and thereby more of carbonic acid will dissociate to regenerate the lost H plus and regain the blood pH. Now, if the carbonic acid dissociates, then more of dissolved carbon dioxide will combine with water to form carbonic acid, and then to regenerate the dissolved carbon dioxide level, more of gaseous carbon dioxide from the lung airspace will enter the blood. Now this is controlled by controlling the rate of breathing, which in turn is controlled by the brainstem. Now normally brain can sense the rise in the acidity of blood, like this lowering of pH, as well as increase in the partial pressure of carbon dioxide. In both cases, it will lead to triggering deeper and more frequent breathing and thereby regulate the blood pH.
Sir, can water directly participate in biochemical processes as a reactant?
There are many such examples where water participates directly in one of the most important biochemical processes of life. Say there are certain reactions called condensation reactions where two molecules condense to form a bigger molecule. Like here ADP combines with inorganic phosphate forming ATP. Here you can see water is eliminated between the two molecules. Now this is an example of condensation reaction. If we extend this example, we will see that when biological macromolecules form, then a lot of monomeric units are polymerized. Nothing but condensation reaction. In each case, water molecules are released. So these type of reactions, the condensation reactions, they are usually endergonic reactions. That is, it needs input of energy to perform this type of reaction. Interestingly, the reverse is also very important. Like water can directly attack a bond and cleave it. Okay, this is called hydrolysis. Even the macromolecules, they are usually depolymerized by this type of hydrolysis to generate smaller components. Now, in contrast to condensation reactions, the hydrolysis reactions, they are always exergonic. That is, they lead to release or formation or generation of energy. Another important type of reaction where water is involved is the oxidation of fuel molecules. When fuel molecules are oxidatively degraded, usually they generate carbon dioxide and water as the end products, whether it be carbohydrate like sugars or glucose or fatty acids. Actually, these type of oxidative degradation of fuels, they can generate a lot of water. And for some animals, this is very important for their lifestyle. For example, camels, which spend a lot of time where they do not drink any water. So the only source of water for their survival is this metabolic water. Then of course we have one of the most important reactions of life, that is photosynthesis, green plants and green algae, which use the energy of sunlight to split water. So in this reaction you have water and you have this species A, which is electron acceptor, where water donates its electron to A. Usually this is a series of oxidation reduction reactions and fundamental to all sorts of life. So overall we can say that we have discussed three important aspects of water in the context of biological processes. First of all, osmolarity. Water determines the osmolarity of intracellular fluid as well as extracellular fluid, and that plays very important role in cellular life. Then we have seen the importance of maintenance of pH within the cell and outside the cell. These are done by different buffer systems, and these buffer systems can only act in aqueous environment, nowhere else. And finally, we have seen water as direct participants in pivotally important biochemical reactions and thereby dictate the properties of life.